Hello, my name is Ilona Kuzmina and I'm from University of Latvia. I will present our study of agar-based phantoms for skin diagnostic imaging. The aim of this study was to validate previously developed smartphone-based RGB imaging devices and algorithm for concentration evaluation of skin chromophore using agar-based phantoms. To achieve the aim, we set the following tasks: to produce one-layer and two-layer agar-based phantoms with different chromophore concentrations, to evaluate absorption properties of the phantoms using transmittance and diffuse reflectance images, to analyze the influence of experimentally determined photon mean free path values on chromophore concentration maps, and to compare the results obtained from RGB images captured by two self-developed smartphone-based prototypes. The agar phantoms have a lot of benefits, which are listed in the slides. They are easy to produce, flexible, biologically compatible. The index of refraction of such phantoms is close to the index of the tissues. Agar hydrogel allows to make different shapes, layered structures, and to make inclusions. The main limitation of these phantoms, they are not stable over a long period of time. We use the following main components to produce the phantoms. Matrix was made from agar powder that was diluted in water and heated. For absorption simulation, we use hemoglobin powder diluted in water. For scattering simulation, intralipid emulsion. This slide shows the different types of agar phantoms we made. We made one-layer phantoms of different thicknesses from 1 to 3.5 centimeters and use these phantoms to calibrate our devices by adding different hemoglobin concentrations. We also made phantoms with hemoglobin inclusions and two-layered phantoms with simulated vascular vessels for skin chromophore mapping. The width of each phantom was 5 centimeters. This slide shows examples of our phantoms. The first one is pure agar phantom. The second and third one are phantoms with the added hemoglobin of different concentrations and thicknesses. The last two are the phantoms with simulated neoplasms and skin vascular vessels. Here are summarized phantom measurements that we performed, and I will describe them more detailed in the next slides. We measured phantom spectra in visible and near-infrared wavelength region, and also measured photon time of flight signals to evaluate photon mean path lengths. RGB images were captured by two smartphone-based devices, LED and laser illuminators. Phantom spectra were measured using halogen lamp and spectrometer at source detector distance of 400 micrometers. The phantoms were placed on a ring approximately 30 centimeters above the surface to eliminate scattering from the background. Remitted photon propagation time, or photon time of flight, was measured by a time-correlated single photon counting system and white laser with broad emission spectrum. The wavelengths were selected by using interference filters. In this study, we measured signals at 520 nanometers and 680 nanometers at source detector distance of 1 millimeter. The measured signals were used for mean path length evaluation, and detailed description of calculation you can find in our papers mentioned below. RGB images were captured by Nexus 5 smartphone camera under subsequent red, green and blue illumination of LED prototype using specially developed smartphone app Skin Viewer. A snapshot RGB images were captured under simultaneous illumination of red, green and blue laser line of laser prototype by AZ camera app. This slide shows the measurement schemes of transmittance and reflectance imaging. Transmittance imaging was performed by placing a phantom on the top of the illuminator, while smartphone was placed at a focus distance from the phantom. Diffuse reflectance images were captured by placing illuminator in contact with the phantom. We evaluated optical properties of the phantoms using B-Lambert law as described in this slide. We calculated absorption and extinction coefficients as well as hemoglobin concentration. The thickness of phantoms was used for calculation from transmittance images and photon mean path lengths for calculations from diffuse reflectance images. In our calculation, instead of incident light intensity, we used transmitted or reflected light of the reference phantom. These tables show detailed description of the hemoglobin and intralipid concentrations used in the one-layered phantoms of different thicknesses. The hemoglobin concentrations were changed from 0.5 to 3.5%. That corresponds to 1.5 to 10.5 micromoles. As reference, we used phantoms of pure agar and agar with intralipid. This slide shows the spectra of the phantoms with different hemoglobin concentrations. In the left graph, the spectra are normalized to white reference, in the right graph to pure agar phantom spectra. As you can see, agar phantom without hemoglobin has also many absorption peaks, we suppose mostly due to water presence in the phantom. Spectra normalized to the spectrum of the pure agar phantom shows the hemoglobin absorption peaks near the 400 nanometers and between 500 and 600 nanometers. This table summarizes the calculated values of photon mean path lengths for phantoms with different concentrations of intralipid and hemoglobin. The mean path length values are in the range from 14 to 24.5 millimeters. We use these results for calculation of phantom concentration maps from diffuse reflectance images. In this graph, you can see the dependencies of absorption coefficients obtained from transmittance images on hemoglobin concentration in the phantom. The results are given for LED and laser prototypes and three different phantom thicknesses: 9 mm, 1.5 cm, and 3.3 cm. The absorption linearly increases with the increment of concentration. In most of the cases, the highest value are observed for images captured under blue illumination at 460 and 448 nanometers. The lowest under red illumination at 660 and 663 nanometers. The exception are thick phantoms where blue light was strongly absorbed and it wasn't possible to evaluate absorption from dark images. Here we compare extinction coefficients achieved from the green and red images of the phantoms with different thicknesses. It's expected that the values of extinction coefficient should be independent on hemoglobin concentrations and phantom thicknesses. The results show the less fluctuations of the values for images captured by laser prototype at 660 nanometers. However, values depends on the phantom thickness. Coefficients obtained from laser prototype are higher for the phantoms with the thickness 9 mm and decrease at greater phantom thicknesses. These graphs compare the mean values of hemoglobin extinction coefficients obtained by LED and laser prototypes with the values presented by other authors. These results are obtained from transmittance images of 9 mm and 1.5 cm thick phantoms. As you can see, our calculated extinction coefficients are higher compared to the values found in the literature. The greater difference is observed at 460 nanometers, and the difference decreases with increasing of the wavelengths. Here we compare hemoglobin extinction coefficients obtained from transmittance and reflectance images with the value found in the literature. The thickness of the phantoms in both cases are the same, 3.3 centimeters. In the case of diffuse reflectance, extinction coefficients were calculated using the experimentally determined photon mean path length. The values obtained by the laser prototype are closer to the reference data compared to the LED prototype. If we compare diffuse reflectance and transmittance measurements, the results from diffuse reflectance images are much closer to the reference data. This slide demonstrates diffuse reflectance RGB image of simulated skin vascular vessel and corresponding chromophore maps calculated from images captured under green and red illumination by LED prototype. This is two-layered phantom, as you can see in the scheme below the maps. Both layers are made from pure agar. The vascular vessels were engraved in the bottom layer, where hemoglobin stock solution also was filled. The jet scale of the maps show hemoglobin concentration in micromoles. Here you can see the illustration of the calculated hemoglobin concentration maps of simulated skin vascular neoplasms. These are one-layered phantoms with inclusions. The matrix of the phantom is made of agar with intralipid. Neoplasms are made of hemoglobin agar solution and hemoglobin water solution. The concentration of the hemoglobin in the hemoglobin agar solution was 6 micromoles, in hemoglobin water solution 307 micromoles. The maps show that the calculated concentration in the first case does not exceed 3 micromoles, and in the second case does not exceed 15 micromoles. To summarize, in this study we presented agar-based phantoms for evaluation and mapping of the hemoglobin concentration by transmittance and diffuse reflectance RGB imaging. We evaluated hemoglobin concentration maps of the phantoms from diffuse reflectance images by using the experimentally measured photon time of light signals. As we know, no one has done it before. If we compare diffuse reflectance and transmittance images, the results show that the values of extinction coefficients from diffuse reflectance images are closer to the values obtained by other authors. And the laser prototype showed more stable and precise results compared to the LED prototype. I would like to thank the European Regional Development Fund for supporting this study. Thank you for your attention, and I will be happy to answer your questions.